All right, good afternoon, everyone. Thanks, Mark, for that introduction. So I feel a little bit like this will be a Monty Python segue, and this is now time for something completely different. So I'm going to take you through a deep dive into pharmacology here, and also talk about Huntington's disease. So before I get into Huntington's disease, by way of disclosures, I am currently collaborating with Cannabinoid Therapeutics on an NSERC CRD grant. That's not related to what I'm presenting today, but it is worth noting. But other than that, I have no conflicts or disclosures to make. So Huntington's disease is an autosomal dominant neurodegenerative disorder. It's caused by the inheritance of a single copy of the mutant Huntington transgene. So if one of your parents has Huntington's disease, you have a 50% chance of getting Huntington's disease yourself. Typically, patients that suffer from this disease will begin to experience motor symptoms in the 3rd to 4th decade of life. Those will include symptoms such as choreiform movements and difficulty swallowing. Prior to the onset of motor symptoms, the patients will also experience changes in behavior and mood. And symptoms tend to last 10 to 15 years until eventually the patient loses control of their swallowing reflex, and typically death is by asphyxiation or choking. There is no current verified treatment for the disease itself. There are treatments that are prescribed to the patients to help control changes in psychoses or in mood and behavior. And there are one approved drug, tetrabenazine, to help control the choreiform movements. There's also some promise recently in gene therapy for Huntington's disease, and those trials are still ongoing. But no disease-modifying agents for Huntington's itself. So one of the first things that happens prior to the onset of motor symptoms and early in changes in mood is that we see a change in gene expression in patients with Huntington's disease. And that change actually occurs through the mutant Huntington protein itself, transcriptionally dysregulating the type 1 cannabinoid receptor. You'll see it on my slides as CB1R, because that's how I just abbreviate it. So this change occurs primarily in the caudate and putamen and the globus pallidus. These are areas that are responsible for motor coordination in the brain. And you can see here on this PET imaging scan, looking at CB1 receptor radioligand binding, that early in disease progression in, I don't think this laser points, but early in disease progression in that middle slide, there's a precipitous drop off. Oh, thank you. There's a precipitous decline in the caudate and putamen, which is right here, compared to the early, and then that remains low. It drops by about 50% in these patients. So in Huntington's disease mouse models, what we see is that mice that are knockouts for CB1 that are crossbred with Huntington's disease mice progress into their Huntington disease symptoms earlier, and they display more severe symptoms, more severe choreiform movements, and more severe changes in memory and behavior. There have also been a number of studies to demonstrate that if you rescue CB1 expression, which is to say you add it back in with an adeno-associated virus, you can restore some of the normal phenotype in these animals that have Huntington's disease. So here's some patient data. So this was from tissue collected from deceased patient donors. And what you can see is that in both grade 3 and grade 4 Huntington's disease patients, CB1 mRNA levels drop precipitously compared to the normal control. And it's thought then that this loss of CB1 is correlated with disease progression. And we hypothesized that if there were a mechanism to restore signaling of CB1 in Huntington's disease, that you might actually be able to reduce some of the signs and symptoms of the disease. So not a cure by any means, but a treatment or a disease-modifying agent. So that was the question we asked, is there a way to maintain function or increase levels of the receptor in Huntington's disease through a pharmacological tool? So there's a lot of different ways that we could do this. We could look at restoring levels of endocannabinoids such as anandamide and 2-AG. And the previous speakers gave a really good outline of the endocannabinome, and so I won't go into that too much, but that's one way we could do it. Unfortunately, it doesn't look like you could use a direct-acting CB1 agonist, because one of the things we would want to avoid is that chronic downregulation or desensitization of receptor. But one way we could approach this is through allosteric modulation. So what's an allosteric modulator, you all ask? This slide, I'm just going to check to see if this is going to work. Oh, excellent. So typically when we think of a G protein coupled receptor like CB1, what we see is that we have this agonist, so we can imagine this is any endocannabinoid, and it binds to the receptor and it promotes some level of signaling. Well, if we have an allosteric agonist or an allosteric modulator, pardon me, on board, we can actually affect the potency and the efficacy of signaling through that receptor. So the allosteric ligand is going to bind to a distinct site somewhere on the receptor, here shown with my pink bubble, and the pink bubble is going to bind to a separate site on the receptor and affect the potency, which I show with alpha, and the efficacy, which I show with beta, and you get more signaling. But all we're doing is enhancing the endogenous tone. We're not actually activating the receptor directly. So in the absence of an orthosteric agonist, no signaling would occur in theory. We also have the reverse, which is a negative allosteric modulator, so binding, again, that same pink bubble site, but now instead we're decreasing endogenous tone and endogenous signaling. But I'm going to focus today on the positive allosteric modulation, so upregulating endogenous signaling. So this is important because in theory we have reduced likelihood for dependence, tolerance, or adverse effects, because the only way the drug acts at the receptor in theory is through the presence of that orthosteric ligand. And today we're going to be talking about that in the context of anandamide or triracidotyl glycerol, our major endogenous cannabinoids. So there already are some CB1 allosteric modulators out there. The two on the top are ORG and PSN-CBAM. Their pharmacology is quite complex. These are synthetic allosteric modulators, and they're thought to be negative allosteric modulators, so compounds that reduce potency and efficacy through the receptor. But they are kind of complicated, and I could spend an hour talking to you about all the different subtleties associated with those. Then we have compounds like lipoxin A4. Lipoxin A4 is an endogenously produced native compound to the body. It's thought to be an unstable, so readily degraded labile compound that acts as a positive allosteric modulator. But because of this instability, it might not be useful to treat animals with it because it is transient in nature. So what we did was we formed a collaboration with this gentleman here, Dr. Ganesh Thakur at Northeastern University, and we began to synthesize more and more allosteric modulators. To date, this number is actually out of date. We've got 360 allosteric modulators that we've developed a library for, and trying to really understand what makes the best positive allosteric modulator or negative allosteric modulator for the receptor. We've found a number of negative allosteric modulators that range in potency, as well as positive allosteric modulators that range from mid to high potency. Many of these compounds themselves display what is called ligand bias. So ligand bias is another pharmacological term, and it refers to this idea that you can target the different signaling pathways that a GPCR can work through selectively. So let's say, for example, you wanted a GPCR compound that only signaled through G proteins but not beta-arrestin, then you could develop a biased compound that would only signal, or preferably signal, through G protein coupling and not through arrestin, and select the type of signal profile that you wanted. And so with these allosteric modulators, we also see bias. And then importantly, all of our experiments get validated right now at least 4 labs. My lab, a lab at Dalhousie University, and then a laboratory at the University of Aberdeen and at Indiana University. And so we're all working together to really screen and understand this new class of compounds that modulate the CB1 receptor. So here is the compound that I'm going to spend most of the rest of my talk discussing. This is GAT or GAT211, and it is a CB1 positive allosteric modulator. GAT has been investigated already for applications in ocular hypertension by Dr. Melanie Kelly's lab, as well as applications for neuropathic pain by Dr. Andrea Homan's lab, and it does seem to have efficacy in both models. And here we're going to talk about it in the context of Huntington's disease. GAT211 is special because it's a racemic compound, and I've highlighted the stereocenter there with the red asterisk. The racemic compound we call GAT211. GAT228 is the R enantiomer, and GAT229 is the S enantiomer. So they're different stereocenters, different chirality. And for the rest of this talk, if you see blue, think R. If you think red, think S. And that's how we'll sort it out just for ease of data. This is a really good example of how you can explore the pharmacology of some of these cannabinoids and kind of not get frustrated, but get into a place where you need to work through some of the intricacies and complexities because what you observe doesn't necessarily align with the theory. So I got a bunch of data from a number of experiments. This is in cultured cells expressing the type 1 cannabinoid receptor. And we're looking at cyclic AMP inhibition. This is a predominant signaling mechanism through G proteins that CB1 works on. And what we did was we treated these cultured cells with the synthetic CB1 agonist CP55940, and then we added on top of that increasing dosages of either GAT211, 228, or 229. So what we saw first was that GAT211, our gray compound, increasing concentrations of that caused an increase in the baseline and a leftward shift. This was puzzling to us because if a compound was truly a positive allosteric modulator, there should be no shift in baseline. A baseline shift indicates that that compound is activating the receptor all on its own, and that's weird. So we separated out the enantiomers. GAT228 did produce that shift in baseline, but no shift in potency or efficacy. And then when we tested GAT229, the S enantiomer, what we saw was a leftward shift in potency and an upward shift in efficacy and no change in baseline. And I was able to work with Dr. Terry Kennecan, who really helped me understand and grasp this complex pharmacological data in order to say that when we separate out the enantiomers, what we see is that that GAT229 compound, that's the true positive allosteric modulator, and the S enantiomer, the R enantiomer, is a mixed compound displaying both allosteric and agonistic properties, which again goes against this idea that everything can be neat and simple. In cannabinoids, nothing is neat and simple. So despite a large body of literature on CB1, we are still only beginning to understand the structure-activity relationships of cannabinoids at their receptor. That's because we have scaffolds from natural sources, but we also have a wide variety of synthetic sources. And it was only last year that the crystal structures for the receptor were actually figured out, and now we can begin to have rational drug design for these receptors. So what we thought was that if we could apply our PAM, GAT229, we could effectively enhance endocannabinoid activity and possibly provide benefit in an animal model of Huntington's disease. And at this point, I want to remind everyone that GAT229 is not a drug. It is a probe compound to demonstrate proof of principle. There are several pieces to this molecule that don't make it a good drug, but it is a proof of principle probe compound that we can use to explore this space. So the first thing that we did was we looked at CB1 mRNA levels to see if we could rescue or restore CB1 in these animals. So working our way from left to right, we see the vehicle-treated animals, they have low levels of CB1 mRNA. If we then go across, we can see that GAT211 and GAT229 both increased CB1 mRNA levels relative to vehicle-treated R62 or Huntington's disease mice. And that was really exciting for us. If we then looked at symptom progression in these animals, so this is a weighted score. We have grad students that are blinded to what the treatment is, and they observe these animals, and they score them on a criteria of 9 different pieces that make up the Huntington's disease phenotype and try to understand how sick these animals are. We go back and we look at them every day while they're being treated with these compounds daily. And what we see is that the vehicle-treated mice in the clear circles, they progress through the disease. So if you look from day 0 to day 21, they get gradually worse in their disease progression. The GAT211 mice, we see that there's a delay until about day 17, at which point they jump up again and they begin to show symptoms. So there's been a delay in symptom progression in these Huntington's disease model mice. The GAT229 treated mice over the course of the study do not develop or progress into an HD-like phenotype. And this was a short trial, and we're now following this up in more long-term models of Huntington's disease that express the mutant huntingtin protein at lower levels. They're less aggressive. The other phenotype that these animals have is that over the course of treatment, not over the course of treatment, over the course of the study, vehicle-treated and normal R62 Huntington's disease mice lose weight. And we wanted to see if there were any sort of metabolic effects to this treatment. And what we saw was that in the case of GAT229 and GAT211, our positive allosteric modulators, we do see a normalization of weight. So the animals that are treated with these compounds appear to improve in terms of weight gain. And then if we look at the fat content of these animals, interestingly, we see that the weight loss that occurs in the Huntington's disease mice is actually skeletal muscle weight loss, and the percent fat increases. So we can think of these mice very much as fat, skinny mice in the sense that their percent fat goes up. But in the case of GAT228, which was our weird compound, we actually saw an exacerbation of that percent fat content, which was interesting. So to wrap it up, I realized that I'm the barrier between you guys and lunch, so I won't keep you too long. What we saw was that these CB1 positive allosteric modulators were able to enhance endocannabinoid signaling, and we could have something that could be developed for use in treating Huntington's disease both within the neuron and between neurons, as well as looking at some of the behavioral and phenotypic signs and symptoms of Huntington's disease and the metabolic profile. What we're hoping now is that now that we have resolved crystal structures for the cannabinoid receptors, we can begin to explore the structure-activity relationship and get a better handle on rational drug design, not just in terms of these synthetic compounds that I've spent my time talking about, but also in terms of things like cannabidiol and THC, and trying to better understand how those compounds naturally bind to the receptor, which is important if we want to understand the endocannabinoid system and my favorite receptor, CB1. So what are we doing now? So these are some of our big questions. I've helped to form a team at the University of Saskatchewan. We're called CRIS, the Cannabinoid Research Initiative of Saskatchewan. We're currently running a trial for children with refractory encephalopathy epilepsy, and we're hoping to expand that trial out. And then we're also looking to develop future clinical projects that you can ask me about later at lunch maybe. We're also looking at developing these positive allosteric modulators for a number of other disease states. One of them that my postdoc is currently working very hard on is in this Gare's rat model of absence epilepsy, as well as in an EAE model of multiple sclerosis. And finally, looking again at those structure-activity relationships to try and better understand what the cannabinoid receptor's ligand binding site looks like, and then applying that knowledge both in cell culture but also in animal models. And then one of our clinical goals is to be able to develop more and newer PET radio tracers to be able to look at cannabinoid receptor binding in the brain and the periphery in human patients. So I have a lot of acknowledgments to make. I very strongly feel that science is a team sport. So in my lab at the U of S, Miriam, John, Andrew Roebuck, and Quinton, Dr. John Howland has been really helpful in getting me situated at the U of S. And he's a good colleague of mine. And then at Dalhousie University, my former supervisor, who's continuing to collaborate with me, as well as Dr. Melanie Kelly and Amina and Jill. And then our many sources of funding that have been very helpful to us, as well as collaborators at Northeastern University and the University of Aberdeen. So thanks so much, everyone, and I think I have time for questions.